Okay, I'm going to go ahead and start. Okay, those of you on Zoom, can you hear me okay?
Yep.
Can you hear me
Yes.
okay? Okay.
Yeah, we can.
All right, good. All right, I'm going to share my screen. Just a minute here. Try this again. Okay, now we got it. Right, today we're going to continue on some of the biochemical topics. And the next one is on the subject of carbohydrates. Right, a carbohydrate is defined as a polyhydroxyaldehyde, polyhydroxy ketone, or a substance that gives these compounds on hydrolysis. Okay, so a polyhydroxyaldehyde or ketone means that you have an aldehyde or ketone functional group, and all of the other carbons will contain an alcohol. There are various classifications of carbohydrates. One is the monosaccharides. These are simple sugars. They are carbohydrates that cannot be hydrolyzed to a simpler carbohydrate. Monosaccharides will have the general formula CN2NON. Okay, and so we have hydrogen and oxygen in a ratio of 2:1, so the name means hydrates of carbon. Okay, and aldose is a monosaccharide in which the principal functional group is an aldehyde. A ketose is one in which the principal functional group is a ketone. Monosaccharides are also classified according to the number of carbon atoms. Okay, using these prefixes, triose is 3, tetrose is 4, pentose is 5, hexose is 6, and so on. Okay, most monosaccharides are 3 to 8 carbons, with 4, 5, and 6 being the most common. Okay, there's only 2 trioses. And the term aldotriose combines the two classifications we were looking at in the previous slides. Okay, aldo, meaning the principal functional groups in aldehyde, and then triose, 3 carbons. Okay, so there's only one aldotriose, and that's glyceraldehyde. Okay, we have an aldehyde on carbon 1 and alcohols on carbons 2 and 3. Okay, there's only one ketotriose, and this is called dihydroxyacetone. Okay, often aldo and keto are omitted, and these compounds are referred to simply as trioses, tetroses, and so forth. Although these modified designations do not tell the nature of the carbonyl group, they at least tell the number of carbons. Okay, so glyceraldehyde has a chiral center. It exists as a pair of enantiomers. Okay, the chiral center here is on carbon number 2. Got 4 different groups: aldehyde, this hydroxymethyl, the OH, and the hydrogen. Back in our chapter on stereochemistry, we introduced the idea of Fischer projections. Fischer projection is a 2-dimensional representation for showing the configuration of a tetrahedral stereocenter. Okay, so horizontal lines represent groups that are going back behind, and horizontal groups are coming forward. Okay, so this Fischer projection corresponds to this 3-dimensional model. Okay, the first and last carbons in the chain are written in full. Any chiral centers are indicated by the crossing of bonds. Okay, so this pair of intersecting lines indicates a chiral center. Right, in 1891, Emil Fischer made the arbitrary assignments of D and L to the enantiomers of glyceraldehyde. Okay, he assumed that this configuration was the D, and it turns out he guessed correctly, and this is the L. Okay, the optical rotation is the same number of degrees, but in opposite directions. Right, according to the conventions proposed by Fischer, a D monosaccharide is a monosaccharide that, when written as a Fischer projection, has the OH of its penultimate carbon on the right. Okay, penultimate next to the last, the highest numbered chiral center. And L would have the OH on that carbon on the left. Okay, so the penultimate carbon is the next to the last one. That's this one right here. And these are all D sugars. Okay, since the OH is on the right. Okay, for 4-carbon aldoses, there are 2 of those. And these are erythros and threose. And there's a mnemonic. Help us remember these. And that's TOES. Okay, this stands for threose, opposite, erythros, same. Okay, so threose has the OHs, the chiral OHs on opposite sides in the Fischer, and erythros has them on the same side. Okay, this is a pentose. There's two of the most common aldopentoses. While there's only 2 of these aldotetroses, there are more than 2 of these. Ribose is one of the most common. This has all the OHs on the same side. And then this is 2-deoxy-D-ribose. Okay, this this OH has been removed. Okay, strictly speaking, this should be written as CH2 since it's not a chiral center. Okay, these are three of the most common aldohexoses. There's actually 8 of these. Okay, we have glucose, galactose, and D-glucosamine. Okay, in D-glucosamine, the OH has been replaced by an amino group at carbon number 2. Okay, these are some amino sugars. We just saw D-glucosamine. This one is D-minosamine and D-galactosamine, and N-acetyl D-glucosamine. N-acetyl D-glucosamine is a component of many polysaccharides, including the connective tissue, such as cartilage. It's also a component of chitin, which is the hard shell-like exoskeleton of lobsters, crab, and shrimp. Right, monosaccharides have hydroxyl and carbonyl groups in the same molecule and exist almost entirely as 5- and 6-membered cyclic hemiacetals. Okay, the anomeric carbon is the hemiacetal carbon of a cyclic form of a monosaccharide. And then anomers are monosaccharides that differ in configuration only at their anomeric carbon. Okay, so anomers are actually a specific type of diastereomer. Okay, so here we have D-glucose. Okay, back in our chapter on aldehydes and ketones, one of the things I mentioned was that most hemiacetals are not stable. It turns out with these 5- and 6-membered rings, the cyclic hemiacetal is actually the favored equilibrium form. Okay, so the carbonyl is polarized, carbon end is plus. The lone pair on the oxygen here is the nucleophile, so that attacks the aldehyde carbonyl carbon. That closes up the ring. We have a proton transfer that occurs, and we get this cyclic hemiacetal. It's a 6-membered ring, heterocyclic ring. Okay, one of the members is an oxygen. Okay, it's a hemiacetal. Definition of a hemiacetal is we have an ether and an alcohol on the same carbon. Okay, well, it turns out that this ring can close from 2 different directions. It can close on one side or the other side, which gives rise to 2 isomers. Okay, where OH is pointed up in this configuration, that's called the beta form. And where it's pointed down, that's the alpha form. And this is the anomeric carbon where the ring closes and where we have this cyclic hemiacetal group. Right. Excuse me. Haworth projections. What we're going to look at next. 5- and 6-membered hemiacetals are represented as planar pentagons and hexagons viewed through the edge. Okay, that's what Haworth projections means. Most commonly, these are written with the anomeric carbon on the right and the hemiacetal oxygen to the back right. Okay, so let's back up. Okay, hemiacetal carbon's on the right, and then the heterocyclic oxygen is back behind. Okay, a 6-membered hemiacetal, and then I mentioned this, if the OH on the anomeric carbon is cis to the terminal CH2OH, then that's beta. I'm sorry, it's alpha, and beta would be trans. Okay, so a 6-membered hemiacetal is shown by the prefix pyran. Okay, it's related to this heterocyclical pyran, and then a 5-membered hemiacetal is shown by the infix furan. Okay. So this is a 5-membered ring, and this was a result of ribose closing up to form this cyclic hemiacetal. So this is alpha D ribofuranose. Okay, the furanose indicates a 5-membered ring. The ose indicates a carbohydrate, monosaccharide. And alpha means that this OH on the anomeric carbon is pointed down. And then this is the beta isomer. The most prevalent forms of D-ribose and other pentoses in the biological world are furanoses. Deoxy means without oxygen, so this one is 2-deoxy D-ribofuranose. Okay, notice the OH is missing. Right, D-fructose, or 2-ketohexose, also forms alpha and beta cyclic hemiacetals. Okay, so this is fructose or fruit sugar. And the ring can close from 2 different directions. Okay, this is the alpha form, where OH is down. This is the beta form where the OH is up. This is the anomeric carbon right here. Right, 5-membered rings are actually close to planar, and Haworth projections are adequate to represent furanoses. Six-membered rings of pyranoses are more accurately represented as chairs, which are strain-free. Okay, so here is beta D-glucopyranose or pyranose. Okay, the ring opens to form glucose. And then when it closes back up, then we have the alpha. Okay, at equilibrium, there is 64% of the beta and 36% of the alpha isomer and less than about 0.5% of the open chain. Okay, so this is in constant equilibrium. The ring is continuously opening and closing, but the favored equilibrium forms are actually the cyclic forms. And you have almost twice as much of the beta as you do the alpha at equilibrium. Okay, so the point of this slide is that the planar hexagon doesn't actually accurately represent the shape of the molecule. Okay, so here's the Haworth and here's the chair. Okay, in glucose, or glucopyranose, I should say, for the beta form, okay, notice the OHs are alternating. This is up, down, up, down, up. CH2 then is up. And for the beta form in the chair, all of the OHs and the CH2 OH is equatorial. Right, so what is mutorotation? Okay, so alpha D glucose has a certain measured optical rotation, and beta D glucose also has a certain optical rotation. And at equilibrium, okay, the measured optical rotation changes. Okay, when it reaches equilibrium, then the measured rotation is 52.7. Okay, and this is one of the ways they can tell what the percentages are at equilibrium. Okay, galactose is another monosaccharide, and it also undergoes mutarotation. And at equilibrium, again, it's mostly the beta. Okay, so beta-D-glucopyranose, this can be, these two different forms can be crystallized. So when we dissolve this, it starts out at 18.7, and it converts to the alpha, and then the alpha converts back to the beta, and until equilibrium is reached. Right, a glycoside is a carbohydrate in which the OH on its anomeric carbon is replaced by an OR, okay, such as a methyl group. A glycosidic bond is a bond from the anomeric carbon to the glycoside of the OR group. Okay, so to name a glycoside, we name the alkyl or aryl group bonded to the oxygen, followed by the name of the carbohydrate, and we replace the ending E by "ide." Okay, so this structure that was pictured on the previous slide would be methyl beta-D-glucopyranoside and methyl alpha D-glucopyranoside. Okay, there are a number of N-glycosides. One group that is particularly important are the bases of DNA and RNA. DNA and RNA have two major differences. One is for DNA, the sugar is deoxyribose, 2-deoxy-D-ribose, whereas in RNA it's ribose, D-ribose. The other difference is that okay, there are 4 bases in DNA. There's cytosine, thymine, adenine, and guanine. In RNA, thymine is replaced with uracil. Okay, so here is the glycoside formed between D-ribofuranose and cytosine. We have this D-glycosidic bond. Okay, next we'll look at some of the reactions of monosaccharides. One of them is reduction. Since these are, after all, aldehydes and ketones, they can be reduced with reducing agents such as sodium borohydride. Sodium borohydride converts aldehydes and ketones to alcohols. Okay, so here we have D-glucose. It actually has, D-glucose has to be in the open chain form. It can be reduced by sodium borohydride. Aldehyde is converted to a primary alcohol, and this is called D-glucitol. It's also called D-sorbitol. D-sorbitol is an artificial sweetener. Okay, here are some other artificial sweeteners. Sorbitol is found in the plant world in many berries and cherries, plums, pears, apples, and seaweed. It's about 60% as sweet as sugar. Okay, so here's erythritol, mannitol, xylitol, and xylitol and mannitol are used in sugarless gums and candies. Okay, so that was reduction. Now let's look at oxidation of aldehydes. Okay, so D-glucopyranose can undergo oxidation at the aldehyde group to a carboxylic acid. Now in basic solution, we would get the carboxylate. There are some enzyme catalyzed oxidations in which the primary alcohol on carbon number 6 gets oxidized to a carboxylic acid. The body uses this glucuronic acid to detoxify foreign alcohols and phenols. These compounds are converted in the liver to glycosides of glucuronic acid and then excreted in the urine. The intravenous anesthetic propofol is converted to the following water-soluble gluconuride and then excreted. L-ascorbic acid, or vitamin C, is synthesized from glucose, both biochemically and industrially. In our chapter on lipids, we looked at some fat-soluble antioxidant vitamins. Vitamin C is a water-soluble antioxidant. Right, so we've talked about monosaccharides. Sucrose is an example of a disaccharide. Sucrose is obtained from the juice of sugarcane and sugar beets. Okay, here's the Hayworth and the chair representations. Okay, so in sucrose, the two anomeric carbons of glucose and fructose are connected by this ether oxygen. Okay, so here's the glucose, and the configuration of the glucose is alpha at the anomeric carbon. Okay, the O is pointing down. For the fructose, it's pointed up. And so it's alpha with respect to glucose and beta with respect to fructose. All right, here's another disaccharide. This is lactose. Lactose is the principal sugar present in milk. It's about 5 to 8% in human milk, 4 to 5% in cow's milk. And glucose is here on the right, but then on the left we have the monosaccharide galactose. Okay, the only difference between glucose and galactose is on carbon number 4. Right, this OH is axial rather than equatorial. And so this is a 1 to 4 linkage. Okay, carbon number 1 on galactose, and it bonds to carbon number 4 on glucose. Both sucrose and lactose have to be broken down into their monosaccharide units before they can be digested. And this is done enzymatically in the body. Okay, maltose is obtained from the juice of sprouted barley and other cereal grains. Okay, it consists of 2 glucose units that are connected 1 to 4, but it's an alpha 1 to 4 glycosidic bond. Okay, so the Glucose on the left is alpha, and it's connected to carbon number 4 of this other glucose. Okay, if you've had malted milk candy like Whoppers, maltose is responsible for the odor and flavor of that type of candy. All right, let's look at blood group substances. Membranes of animal plasma cells have large numbers of relatively small carbohydrates. These membrane-bound carbohydrates are part of the mechanism by which the cell types recognize each other. They act as antigen determinants. Okay, among the first discovered of these antigenic determinants were the blood group substances. Okay, in the ABO system, individuals are classified according to 4 blood types, A, B, AB, and O. Okay, at the cellular level, the biochemical basis for this classification is a group of relatively small membrane-bound carbohydrates. Okay, one of these membrane-bound monosaccharides is L-fucose. Okay, so here's the difference in blood types. Okay, it's a matter of what carbohydrates are bound to the surface. Okay, so type A has N-acetyl D-galactosamine bonded to it. Type B has D-galactose, and type O lacks these carbohydrates. All right, if we take the structure of maltose, which we saw earlier, that was a beta 1 to 4 linkage of, an alpha 1 to 4 linkage of 2 glucose units. And if we repeat that thousands of times, then we have a polysaccharide called starch. And starch is used for the energy storage in plants. It can be separated into 2 fractions, amylose and amylopectin. Amylose is composed of unbranched chains of up to 4,000 D-glucose units joined by alpha 1 to 4 glycosidic bonds. Okay, all of the linkages are alpha 1 to 4. Amylopectin, on the other hand, is a highly branched polymer of D-glucose. The chains consist of 24 to 30 units of D-glucose joined by a 1 to 4 glycosidic bond, and branch is created by alpha 1 to 6. Okay, so this is what the amylose portion would look like. It would be linear, and then amylopectin has a number of these branches off carbon number 6. Glycogen is also a polymer of glucose, and it consists of alpha 1 to 4 and alpha 1 to 6 glycosidic bonds. Okay, this is the reserve carbohydrate or the storage carbohydrate for animals. The total amount of glycogen in the body of a well-nourished adult is about 350 grams. And it's divided almost equally between liver and muscle. Okay, so the amylose portion of starch had all alpha 1 to 4 linkages. If we have a polymer of all beta linkages, 1 to 4 of glucose units, then we have a polysaccharide called cellulose. Cellulose has an average molecular weight of about 400,000. And that corresponds to about 2,800 D-glucose units per molecule. Both rayon and acetate rayon are made from chemically modified cellulose. Okay, in other words, we put different groups on these OH groups. Okay, I'm going to skip over this. Okay, so here we have, okay, this would be galactose. Okay, well, I can tell that because this OH on number 4 is axial. Right, this is glucose. We have an alpha 1 to 4 glycosidic bond. And here we have a beta 1 to 4 glycosidic bond with another glucose. Okay. Might mention one other difference between starch and cellulose. Okay, starch is the energy storage molecule in plants, and using the enzyme amylase, starch can be broken down into glucose and utilized for energy. Amylase is produced in the saliva and also by the pancreas. So digestion of starch actually begins in the mouth. Cellulose, on the other hand, is not digestible by humans. Okay, because the shape of the molecule is different, the enzymes don't recognize the 1 to 4 linkages, and so we cannot break down cellulose, and it is dietary fiber. Okay, there are some organisms that can digest cellulose. The ruminant animals, cows, sheep, camels, and horses. Well, cows, sheep, camels, horses can also digest cellulose. And they're able to do that because of a symbiotic relationship with a microorganism that lives in their gut. Termites can also digest cellulose, and that's again because of a microorganism that lives in their digestive system. And then wood rotting fungi also produce the enzyme that can break down cellulose. Okay, so I'm going to go ahead and stop here, and I will see you on Wednesday.